Hi, my name is Takeshi Obayashi. I'm an associate professor at Tohoku University. The motivation of this study is how to use gene co-expression information to predict gene function. So this is a typical image of gene co-expression information, namely a gene co-expression network. So these nodes are genes and the edges are co-expression relationships or co-expression distance. Let's suppose that these yellow genes are genes in a target pathway, and now we'd like to find a new gene function as a target pathway. So gene A and gene B is candidate gene, and these genes have co-expression distances to the target pathway like this. So the question here is, which of gene A and B is more strongly associated with the target pathway, and how we can calculate the association index or association degree to the target pathway. We've developed gene co-expression databases for 20 years. In this study, we use the latest version of mouse co-expression data. So this mouse co-expression data has the highest quality in these databases. The target pathway in this study is lysosome. So according to the cake pathway information, so there are 123 lysosome genes in mouse. So we divide it into two groups. So 61 genes are used as target pathway gene to calculate association index, and the remaining genes are used for evaluation of the prediction. We use two types of association indexes. One is distance-based index, and the other is enrichment-based index. So in distance-based index, we directly use the co-expression distance. So the first row indicates the most nearest neighbor, first nearest neighbor target gene from gene A and gene B. And this is the second nearest neighbor target gene and the third nearest neighbor target gene and so on. So when we use distance to the second nearest neighbor target gene, so that is 40 for gene A and 25 for gene B. That means so gene B is more prominent candidate to function in lysosome pathway. On the other hand, in enrichment-based index, so that uses relative position in pathway genes against the other genes. So this is first nearest neighbor gene, second nearest neighbor gene, and so on. And first nearest neighbor gene from gene A is the target pathway gene. Now second and third is not target pathway genes. And then fourth and fifth nearest neighbor gene is again target pathway genes, and so on. So when we use Fisher exact test, so that uses the number of target pathway gene in the given range. So for example, so when we check the top 8 gene in this table, so gene A include 4 target pathway genes, and gene B include 3 target pathway genes. So that means gene A is more prominent as a lysosome pathway gene. In total, we checked 4 types of association index. So 2 of them is distance-based index, and the other one is enrichment-based index. So among these 4 indexes, so the Fisher exact test shows the highest performance. So that means Fisher exact test only uses relative position in co-expression degree. So probably the rank of co-expression strength may be more useful than the co-expression value itself. Thanks.